Okay, thank you. I understand the person holding you from dinner, and we're already running 15 minutes late. So, what I want to do today is to introduce you to technology that we've been developing actually for the last 15 years that is beginning to show some progress, not only in our ability to reach in and turn genes on and off, but actually to modify genes in a very specific fashion. And these are the types of questions that we hope to address. One is, can we create a new approach to therapy using this genetic approach that might allow us to regulate genes in a very specific fashion? And then finally, can we reach in a very specific way, rewrite the genetic script, perhaps to our own benefit? Now, if one wants to consider reaching into the genome, one has to, with a protein at least, one has to consider that protein should be specific so that it should bind the desired site you're interested in and not bind the other 3 billion base pairs of DNA within your genome. So you need a protein, at least in our approach, to read out the chemical identity of each of these bases across a very long sequence. And I show here an 18 base pair sequence that we have successfully addressed. Now, we did this taking insight from nature's repertoire of DNA binding proteins, and what you can see here is that there's one unique protein in this panel, which is a class of proteins actually discovered here at Cambridge University by a fellow named Aaron Klug in the mid-80s, and he coined the name for these proteins, which are zinc finger proteins. You can see they lack the homodimeric nature of most natural DNA binding proteins. That means that they can bind long asymmetric sequences, not sequences that have a symmetry element posed on them, or a palindromic sequence for those of you who do molecular biology. Now, that the other feature that was striking about this protein, not only its lack of symmetry, was that it consisted of 3 modular domains, and this is actually a crystal structure taken from Carl Pavo's work on a related transcription factor. So these modules allow the protein to creep around the major groove in a relatively regular fashion, and that led us to envision that we could perhaps take this natural protein and extend it by several more modules, creating a novel, in this case, 6-finger protein containing 6 of these domains that would allow us to bind 18 base pairs of sequence in a specific fashion. Now, in terms of molecular engineering, this domain is really quite ideal, primarily because of its simplicity. It chelates a single zinc ion. It looks somewhat like a finger, hence the name zinc finger protein as coined by Aaron Klug. It's conserved from yeast to man, and it's the most predominant class of transcription factors in higher organisms. Now, in terms of molecular engineering, it's interesting to work with this protein because it reads out DNA really at a very small portion of that 30 amino acid sequence right here at what I call the reading head of the protein. 1 residue before the start of the alpha helix and 6 residues in allow it to read out a triplet or a codon of DNA sequence. Now, if we want to use these in a synthetic fashion, in other words, we want to be able to build these modules and have them bind any given sequence. Nature has actually only given us a few of these sorts of domains that all actually bind G and N type codons. What we've done over the years is actually to build basically a protein complement of the genetic code. So for each triplet of DNA sequence, now we have a predefined zinc finger domain that will bind that triplet of sequence, and we did that using a combination of an approach called phage display, an approach that I actually presented here about 16 years ago on this very stage, and at least Jim Laird was in attendance at that time, but which we later used to evolve these proteins. And this approach allows you to display billions of copies of a protein on the surface of the virus and then selectively enrich for that protein. About half of those 64 codons came from direct selection. The rest came from design-based studies using the output of this applied molecular evolution. Now what that means, if one has these domains and they're truly modular, you can take these simple domains and treat them like Lego building blocks to create proteins that bind really any given sequence. So if you have 3 such domains, and you can build a 3-finger protein that binds 9 base pairs of sequence, you can easily build 27 such factors. Here's a view of 80 such proteins constructed by simply taking these modular building blocks and putting them together in different fashions. The diagonal represents a unique 9 base pair sequence now that these proteins are binding to with high degrees of specificity. Usually if there's an off-sequence binding, those sequences are highly related. The technique is not perfect, but typically more than 90% of the proteins you build using these predefined domains are functional. It took us a number of years actually to get a crystal structure of one of these 6-finger proteins, and it did indeed look very much like the model that we constructed back in 1997. Now, the first test of the fidelity of this approach was in gene regulation. We looked at 2 tyrosine kinases implicated in breast and other cancers. Found a sequence that was very close within these 2 genes. They actually only differ by 3 base pairs out of 18 in sequence, and then asked if our approach, when we deliver these proteins into a cell, could actually distinguish between these sites and regulate these genes. And so in higher organisms, DNA binding proteins need effector domains to impart regulation. So we applied either a crab domain or an activation domain, for example, to the protein that was designed to bind the genomic sequence of ERB2, delivered those using retroviruses to cell line. And what you can see in this FACS profile is that the transcription factor, that DNA binding protein, with a repression domain, infects those cells and takes the level of protein expression in those cells down to the level of background here. This is the native level, this is the background, and these are uninfected cells. These are the cells prior to treatment. Now, if you just change that repression domain into an activation domain, you see an outgrowth in the FACS experiment. So these cells are expressing more protein on their surface, and we've activated that gene selectively. As compared to ERB3, we make that corresponding factor now, add the repression domain again, we repress down to the level of background. Now the cells don't express ERB3 on their cell surface. With the repression domain, add an activation domain, we see an outgrowth of expressed protein on those cell surfaces. Over the years, we thought when we originally embarked on this program that we would be developing new gene drugs. So these were the drugs that gene therapy had promised us could be used for the treatment of disease, and so we've developed a number of these for a variety of different disease targets. This is one that targets fetal hemoglobin for the treatment of sickle cell anemia. And the cells actually turn pink after delivery of a virus expressing that designed transcription factor that specifically turns on fetal hemoglobin. This is another one designed to bind a relatively conserved element within HIV. These are primary human PBMCs infected with HIV over time. This is the infection growing. These are cells that have been treated with our transcription factor that attacks HIV and suppresses HIV replication. These are the untreated cells. So we can make all of these very interesting transcription factors, and this technology was licensed in the 90s to a company called Sangamo, which now has a drug of this type in the clinic. However, that drug, which activates VEGF, is actually administered by naked DNA injection. So it's a local delivery of a plasmid to a site to affect a therapeutic outcome, and those studies are now in phase 2. This type of approach in terms of building proteins that bind DNAs in a specific fashion has now been automated by us. If you go to our website, you can simply type in a DNA sequence, push a button, and the server generates a sequence of a protein that will bind that sequence with good specificity and affinity, at least good enough to make transcription factors that will allow you to turn those genes on or off. Now, one of the problems with gene therapy is that despite many decades of hype, we really don't have the means of delivering genes in a very safe and effective way into the genome to correct genetic diseases. And so we would like to now use the same sort of technology to overcome this problem. And ideally, as